Hello guys, welcome to my YouTube channel. Today I have a new topic which I want to talk to you, that is centrifugation machine. So what is the centrifugation? First we have to know, then we will be knowing about what is the centrifugation machine useful in which place, or what is the usefulness of the centrifugation machine. So centrifugation machine is a machine, or it's a centrifugation is a process where we can separate the heavy part from the non-happy part or from the lighter part. So if we need an example for this is that how we separate the milk to make curd. First, what happens in the centrifugation machine, what happens, the lighter particles of the milk are in the settle down, the heavy particles get separated into another pan. So like this process is going on. So today we want to learn that how the centrifugation helps in blood banks. It mostly helps to separate the red blood cell, white blood cells, then plasma, then platelets. All these are separated through this process. So because they are not soluble, they are very hard, they are very difficult to separate. So this uses centrifugation process. So there is another question that I want to ask to you, that do you know that what is the purpose of centrifugation machine in blood sample? In laboratories, we were performing a biochemical analysis on body fluids. So centrifuges are routinely used to separate blood cells from serum or plasma, to separate sediment from urine, to measure the volume fraction of erythrocytes in blood. It also known as the hematocrit, and to separate bound from free components in protein. So this is mostly used for the centrifugation. So there is another question that I want to ask, but it's not possible then. So the question is that what is a centrifuge used for in a lab? Why it is used in the lab? Because when we spin at high speed, a centrifuge uses g-force to separate particulates in suspension according to the density. A laboratory centrifuge may be used for sedimentation of cells and viruses, or to isolate macromolecules such as DNA. So this is the main function why we may use the centrifugal machine in a lab. So there is another question what I want to share with you, that what is the main principle on which centrifugation works? Why it is mostly used in blood banks? Because a centrifugal works by using the principle of sedimentation under the influence of gravitational force, which it mostly uses to separate according to the density. The different types of separation including the isopycnic, ultrafiltration, then density, then gradient, then phase separation and pelleting. So these are the main functions. So now what we learn is where to know that this centrifugal machine is used like in the blood bank. So now I want to show you a short video on which we will learn to know more about the centrifugal machine, which is mostly used in the blood bank. So let's see the video.
Topic: Centrifugation. How is the principle of centrifugation helpful in blood banks? Don't know the answer? No worries. We shall find the solution.
Oh yeah! Yes!
Do you know that jelly is a colloidal solution? A colloidal solution is a solution whose particles neither dissolve nor settle down. Another example of a colloidal solution is blood. Blood consists of various components like red blood cells, white blood cells, platelets, plasma, etc., which neither dissolve nor settle down. Usually in blood banks, when a blood sample needs to be tested, its components have to be separated. So, how can we separate those components? What do you think? Can we use a filtration technique to separate various components of blood? No. Filtration is not useful because the size of the components of blood is so small that they can easily pass through the filter paper. Similar to this merry-go-round, a machine called centrifuge is used in blood banks to separate blood components. In the centrifuge machine, a test tube filled with a blood sample is rotated at a constant high speed. This process is called centrifugation. Centrifugation is based on centripetal acceleration. Due to centripetal acceleration, the heavier components, that is, the red blood cells, move outwards. The white blood cells and platelets, which are lighter than red blood cells, form the middle layer, and plasma, being the lightest, moves towards the center. In this way, components of blood get separated by the principle of centrifugation.
After seeing this video, we have now a great knowledge about how centrifugation machine is mostly used in blood banks. So my speech is now completed. So to learn or to know more things, please like, subscribe and follow my channel and press the bell icon to be notified firstly when I upload my video. Thank you.